Have you ever asked yourself how biochemistry helps genetically modified crops and for improved yields, disease resistance, and stress tolerance? Well, we're gonna answer that question. Good morning, everyone. I am Macarla Sihanya and Cebuan. In this video, we will explore how biochemistry revolutionizes the way we grow crops, making them productive, resilient, and nutritious.
So may buntag sa tanan na karon sa Musuan diri sa Basakan sa College Park og naatay sa kamaguma diri nga na problema sa iyang palayan. So may buntag sir.
Ay, magbuntag mo.
So kanang unsay imong kanunay ginaproblema sa imong palayan karon sir?
Thank you for that question. Before that, kanang shout out sa akong boarding house, Ardines boarding house. Hello. Oh my God. Kanang ma'am, ako yung problema diri ma'am kay ako ang mga palay nagkasakit unya nagresulta siya kanang low yields. Unya mugi na akong problema karon ma'am.
Sir ay naami online convention sir ba? Unya would you mind joining us sir para ma-educate ka ug masolusyonan pud imong problema sir?
Hala, ganahan lagi ko anak, ma'am. Sige daw, pa-appeal ko.
So first, we have the biochemical basis of crop improvement. At the heart of crop yield lies photosynthesis. This process converts sunlight, water, and carbon dioxide or CO2 into sugars, which is the plant's energy source. We're studying the enzyme involved in this process like Rubisco to find ways to boost their efficiency. We also investigate how plants manage nitrogen fixation or carbohydrate metabolism and nutrient uptake, because the better the plant is at these functions results to significant increase in crop yields. For example, we have the enzyme Rubisco, which plays a critical role in carbon fixation during photosynthesis. Researchers are working to engineer Rubisco variants that are more efficient at capturing carbon dioxide, which could significantly boost crop yields.
Good day everyone, I am Jules Jen Sivlia and one of the guest speakers for today's webinar series. So I'm going to talk about biochemistry in terms of gene modification for improved yields. So genetic modification involves altering the DNA of crops to introduce desirable traits. This is achieved through biochemistry, techniques like PCR and gene editing tools such as CRISPR. By identifying and isolating genes that enhance growth of nutrient uptakes, scientists can insert these genes into crops, leading to improved yields. Now let's look at some examples. Researchers have genetically modified corn to increase yields by up to 10% by altering the genes that enhances plant growth. Similarly, CRISPR has been used to increase grain yields in maize and rice by about 10% and 8% respectively, without negative effects. Another example is the introduction of genes for enhanced nutrients uptake, which helps plants thrive in challenging conditions. The science behind this yield increase involves altering plant architecture and improving nutrient uptake. For instance, modifying the structure of plants can allow better sunlight absorption and more efficient water use. Additionally, introducing genes that enhance nutrient uptake enables crops to thrive even in poor soil conditions, leading to higher yields. Now, genetic modification powered by biochemistry is revolutionizing agriculture by boosting crop yields. As technology advances, we can expect even more innovative solutions to feed this world sustainably.
Hi, my name is Kanerbs Tiwayan, and I am one of the guest speakers for today's webinar series. Now let's talk about biochemistry in terms of plant disease resistance. Plants like us face constant threats from pathogens, but their defense system operates on a biochemical level. When a pathogen attacks, the plant recognizes it through specific molecules, triggering a cascade of reactions. This involves producing antimicrobial compounds like phytoalexins, which directly attacks these invaders. It also strengthens the cell walls, creating a physical barrier. These responses are driven by complex biochemical pathways. Genetic modification allows us to enhance this resistance. We can introduce genes that code for stronger antimicrobial proteins or thicker cell walls, targeting specific diseases. This can dramatically reduce crop losses, leading to increased yields and less reliance on chemical pesticides. The benefits are clear: healthier crops and reduced chemical use. However, potential drawbacks includes the risk of creating new resistant pathogens and concerns about reduced biodiversity. It's a powerful tool, but careful consideration and ongoing research are crucial for its sustainable application. Thank you for listening to my talk. That would be all.
Hi everyone, my name is Jessica Mariscal Hermosa and I'm one of the guest speakers of this presentation. So, plants often face harsh conditions like drought, heat, and salty soil, which can reduce their growth and crop yield. To help them survive, scientists use biochemical manipulation. They have discovered genes that produce osmoprotectant, which a natural compound that help plant retain water. By adding these genes to crop, plant become more drought resistant. Another method that focuses on controlling how plants use water. So plants lose water through tiny spores called stomata. Scientists can modify this to help plants conserve water without stopping photosynthesis. And by that, it is important in plants because it helps crops survive and grow in harsh conditions. It also enhances their natural stress response through genetic modification. We can improve food security, increase agricultural productions, and support farming facing climate changes. And this innovation ensures that we can continue to produce enough food even if the environment's conditions become more extreme. And that's all. Thank you for listening.